Okay, so the topic for today's class is heredity and evolution. So over here, the part of evolution is cut, so we'll only be looking at the heredity part of this chapter because evolution has been cut from this year's syllabus. Okay, so 1 second. Yeah. Okay, so first of all, everyone can see the screen, right? Yeah, the screen is visible,
Yeah, I
right?
can see.
Okay, fine. Then. So first of all, the term heredity itself. So heredity is the transfer of traits from the parents to their offspring. So yeah, that's the definition of heredity. Now variations, the differences between the characters or traits among individuals of the same species are called variations. Now we've already studied this chapter, so we should be having an idea of it. So now if we're talking about the case, everyone remembers there was an example where a yellow wrinkled seed was crossed with a green round seed. So over here, yellow, 1 second, the color is not okay. So here yellow and so let's denote, first of all, we'll denote the dominant traits with capital letter. So over here, yellow is the capital Y is the denotes yellow and small y denotes green. And the other character was round. Round is the dominant trait and small r, it is the wrinkled. So this property is wrinkled. Now over here, in this, you remember that in the first generation, the F1 generation, the progeny that now progeny is basically the offsprings of when you cross the this part and this part. No, this is not so basically we are crossing this. 1 second. Yellow round seeds and uh, ring wrinkled green seeds. So remember, when you cross this, you get RRYY. That is capital R and capital Y. Basically, you get two dominant traits. So this is basically round and yellow. And so there's no variation here because the parents are round and yellow and also the progeny is round and yellow. So there's no variation here, but the variation comes when you self this. That is, you basically it's basically called selfing. So when you self the parents, so you have another one over here, y. So then you will be getting two extra traits. So over here there was round yellow. After, when you do the F2 generation, you will also get wrinkled green and wrinkled yellow and round green. So these are the new traits you get. So there is this variation that occurs. So the differences between the characters of traits among individuals, it's called variations. Now variations you can see more in human beings. We'll be talking about all that, like why the variations are more in human beings. But if you're giving just an example, suppose if a father, like some random person, so this person, he has black hair, but then his son like has a bit more towards the brown side. So this is just like a small variation that is occurring when the two what do you say, gametes fuse. So we'll be talking about all that. So just keep in mind that the transfer of characters or traits from parents to their offspring is called heredity, and the differences between the characters or traits among individuals of the same species is variations. So now there are a couple of terms that we should be knowing before this. So we'll come back to the importance. So the terms are, first of all, genotype. Now the genotype is basically the genetic constitution of the organism, or basically, if I'm writing like an example, okay. So this is a genotype. This is basically tall tall. So another example where we'll be looking at later, when you cross a tall plant with a short plant, you basically in the first generation you get this. So this is the genotype. Genes, like basically when these two genes, this separates into capital T and small t, so the genes recombine to form this. So this is basically the genotype. Now phenotype, it is the appearance of the organism. So basically it's the outlook of the organism. So the phenotype of the this parent is tall, this parent is dwarf because it's small t, right? So it's recessive. So this is small and this is tall. So this character of dwarf or small, that's what is a phenotype basically. So here example is given. Red color may be controlled by a pair of genes RR. So RR controls the red color of genes, and the genotype. Okay, so if there is RR, so let's just take this one. Now in RR, this is the genotype. This capital R is genotype, and the phenotype is red color. So that's basically what phenotype is. And the next one is gene. So gene is the section of like DNA that determines a particular trait. So all these characteristics that we talk about is basically determined by genes. So it provides information for one protein. And we'll be looking at that. So we'll be looking at how genes control traits, and we'll be looking at that later. But just keep a small idea of what these are. We'll be coming through all this later on in this topic. More terms. So chromosomes are long threads. So basically, our DNA, the term we talk about DNA, it's basically present in chromosomes. So human beings have 46 pair of chromosomes. So 23 pair of chromosomes. So total 46 pairs. So each of these 46 chromosomes have DNA in them, and each of these, like each single strand of these chromosomes have multiple genes in them. It's not just one, it has multiple, and each of these chromosome, it's responsible for a certain character, like not basically character but trait. So we have one pair of sex chromosomes, that is the 23rd pair, and it's only responsible for all these related terms, like our adolescence, like all this kind of stuff that's taking place in our body related to that is included, like the characteristics are included in the 23rd pair. So each pair, like it does not change actually. Like the 23rd pair for all of us, it's like for all human beings, it controls the same characteristic. So just like that, there are 23 different pairs, and it's responsible for different traits. So yeah, and each gene set is present as independent pieces, and this is called chromosome. Now next is allele. So this is a new term. So allele is an alternate form of DNA which determines a trait. So yeah, we'll be coming through all that. Also expressing allele of a gene. Now there is two different types of ways that you can find uh, an allele combination. So let's see. So we just considered this example just a while ago. So we got to know that this pairs, like this forms a tall plant, and this is a dwarf plant because this is dominant. Like when you have two capital letters, first of all, capital letters always determine dominant traits, and small letters always determine recessive traits. Now dominant traits, it can express itself even when recessive traits are there. So over here, if I write, if the first generation forms this, how much ever, like even if you have 1,000 of these, each one of them will be tall because T is dominant. So now there are two different types of combinations that can occur. So first of all, we have these two. One second. This is one, this is one, this is two, and this is three. So these are the only possibilities for it to happen, like only possible combinations, because you can try it out yourself. If like when you take consider the letters tall and like dwarf, the capital T and small t, you can only make three combinations. Now if now homozygous means both are same, like what I mean is that both letters will be same, that is the conditions will be same. So over here you can see it's capital T, and the second one is small t. So both of these are homozygous because um like both of them has the same what do you say, allele basically, and it will always form the trait it has in it. Like for example, small t represents dwarfness. So since both of them are small t, it will form what small t represents, that is dwarfness. Now another thing is that the recessive traits can only express itself in homozygous conditions, that is when it has both of them as small t, or else it will be like the effect of the small what do you say, uh, the recessive traits will be, like it will be kept aside by the dominant traits because they can express itself even when the recessive traits are around. And another term is hybrid. So when you cross a tall plant, that is capital T and dwarf plant, you get small capital T small t. So this is called the hybrid plant first of all, and it's hybrid tall. So yeah, we'll be going through all this just to give you an idea of the terms. We all need to know about this before this. And yeah, we discussed about dominant allele and recessive allele. So dominant allele is always represented by capital letters, and recessive allele is represented by small letters. Okay. Okay, let's just move on. Okay, till here, is there any doubt like in the terms, and is there any doubt in the terms and stuff? If there's any doubt, you can tell, like in the terms and all. Is there any doubt? Because we'll be using that when like in the forward for the crosses and stuff. No doubt, right?
No.
Okay, fine. So the next topic is accumulation of variation during reproduction. So we learned in the last chapter, that last biology chapter, reproduction, that when you reproduce in the sexual method, it produces more variations. So this is because genetic recombination takes place. So what this term is, I'll just try to explain it. So basically, in the asexually reproducing plants, or not just plants, organisms, only one parent is involved. So basically, the same genotype, like the same genes, is transferred. Even though, like the only variations that occur is because of the small, like condition changes, like the changes in condition that occurs while the chemical reactions to form the next generation takes place. But in while in the sexually reproducing organisms, the offspring is like they're receiving two different sets of genes. Over here we saw that yeah. So over here the parent is tall, and over here the parent is dwarf. Okay, so when you cross this, they basically form. This is not how we cross, but just to like show it for now. So you have two offsprings like this. Okay, so both of them have, now if you look at their genotype is, you just, this is one genotype and this is the second genotype. It's completely different from the parent. So this is one variation that has occurred. Now if you self them, you will again get the parental. So first of all, the parental, this parental genotype has occurred again, but you will also notice that there is small t, capital T, capital T, capital T, small t, and one more of this. Okay, there's one more. So you can see the four. There are three different genotypes that occurred from only two, and you can see it from here. Even in the first generation, you're getting that capital T, small t. So this is a kind of a change that has occurred. So you can see that it is receiving one capital T, this one capital T, and one small t. So these two are from two different parents. Okay, so since these are from two different parents, each of them has its own characteristics, like traits. Like I told about the chromosome structure, so I also informed that each chromosome has many genes, and each gene is responsible for one function. So two different characteristics are fusing together to form the offspring, so it brings in more variations. And I told about the, in asexually reproducing organisms, there are changes that occur due to the like fluctuations in the biochemical reactions. Even that happens in sexual reproduction, but only to a minor extent, not too much, but then it happens. But then it's more variations in these organisms are mostly because of the fusing of genotypes, the two different organisms. Like when the genes fuse, that's the reason why more variations are produced. Okay, so uh, okay, now advantages of variations. So basically what we talked about, so if there is a change in the environment, like um maybe some disease, we are facing some a similar situation right now, variations might create, or what do you say, a variation that can make us disease resistant. Like to that, it's really rare though. Okay, it happens in the process of evolution, but then it's really it's time consuming. But you have to understand the advantage of this. So if there is a population of 1 lakh in the world right now, and suppose if only maybe 1,000 of them, like 1,000 of them, or in the beginning we just say 100 of them have gotten this disease resistance character, just assuming, it's obviously you won't get it like that. But then now if these 100 have it, then these 100 can, like when they participate in reproduction, then their offsprings will also be disease resistant. That is, if you're talking about corona, they'll be resistant to corona basically. So these 100 will maybe form 200, 200, it'll grow on to 400, 800, and it just keeps going on. So basically a new set of population is produced that is resistant to corona. And another part of this is that the other organisms will die, like they won't, or what do you say, have this character in them, like this disease resistant. But then the population won't fade away, because there is this 800 or this growing population which is resistant to corona. So it helps in the stability of population of species, and it also helps the organisms to adjust to the changes in the environment. Same example that we have used, and the other organisms. So it's not beneficial to all organisms, as the one without variations will die and become extinct. But they won't become extinct because the variants will survive and they will reproduce and create new organisms like their offsprings, which are also having the same useful variation. Now, variations can also be bad, but then we're not considering that right now. Okay, so that was variations. Now, one second. Pause there. Yes. Oh God. Okay. Yeah, some more advantages. It variation forms the basis of heredity. It leads to evolution and all that. It even does that. So I think we have stated 5 different advantages. So okay, now we get to the main part of this chapter where we are talking about crosses. So there are 2 different types of crosses. The first one is called monohybrid cross. Now, monohybrid cross is where you are only considering one character. We're taking this example again. So basically, this is the most important example basically. So we'll be taking that again. So over here, you're only considering one character, tallness, dwarfness. So basically, you can tallness and dwarfness. They are paired under one category, that is height. So height is the only character that you're looking at in this cross. So you're only considering one pair of contrasting characters in monohybrid cross. So that is their characteristic. Again, you can see the example at a cross between tall and dwarf pea plants. Now for dihybrid cross, we have mentioned another example. Yeah, when you're crossing a round, one second. Yeah, a round green plant with a wrinkled yellow plant. Over here you can see that there are 2 different pairs of characters, 4 contrasting traits, but then 2 different pairs of contrasting traits. This is one, and this is basically, this is round. Okay, this one is round and this is wrinkled. So this is the shape, and this this represents green, and the capital ones are yellow, and this is the color of um what do you say, the offspring's pretty, like, no, not the offspring, the color of the organism that is taking part. So it's basically the trait that it represents. So these 2 represent color and this represents shape. So you have 2 different contrasting character, 2 pairs of different contrasting characters in dihybrid cross. And example is the same thing. Okay, so one second. Yeah, that is the difference. Now, so now we move on to dihybrid, no, monohybrid cross. Okay, so one second, I'm taking the clear slide. So we're doing the same example again, but this time we'll do it properly, okay? So monohybrid cross basically means um when you're crossing 2 organisms with contrasting, one pair of contrasting characters. So over here, if we take, uh, so we're crossing between a tall pea plant and a dwarf pea plant. So this is an experiment done by Gregor Mendel. So he's basically he's called the father of heredity because he's the one that started the, what do you say, all this, his basically the activities related to heredity and all were started by him. And he did this in a time where genes and all were not there, like it was not even there, but still he has found a way to um, investigate on this and learn about it. He called them forms basically. So yeah, one second. Okay, so if there is a father and mother plant, the father will, in this case, will be taking the father to have the tall plant, and the mother will be having the dwarf plant. Okay, so this is the phenotype. So we have talked about the phenotype, the outlook of the organism, or how the organism looks. Like basically, its external character. It's tall and dwarf here, and the genotype will be the homozygous tall and homozygous dwarf. Capital T, and small t, small t. So now this produces two different gametes, capital T over here, both are capital. So you'll only get one type of gamete from here, and even this will give one type of gamete. So both of this will form capital T, small t, because the capital T gamete from here and the small t gamete from here goes and makes one separate organism, that is the F1 generation. So this is the progeny that is producing the F1 generation. Now, so this, okay, so can anyone tell me what is the phenotype of this? What is the phenotype of this? Kedarnath, can you tell? What is the phenotype of this F1 generation that is formed? Yeah, is it tall or dwarf? That's the only like possibility. So is it tall or dwarf?
Which is the majority one?
You can see that there's the capital letter over here and the small letter. So you know that you have to check what the like. First of all, if you're looking at the phenotype, only the tall gene basically. No, the tall part of the genotype will show its characters because it is the dominant trait. So what is capital T over here?
Then it's tall.
Yes, that is tall. This is tall. So the offspring formed in the first generation will be tall because it is capital T is the dominant trait. Now, if you're looking at a pattern that you see in monohybrid cross, the dominant trait is always expressed in the first generation. It's not possible for the recessive trait to express itself in the first generation. So you can keep that in mind. So here, all the offsprings will be tall. So now you're selfing the F1 generation. So you'll be crossing this twice. So you'll be crossing plants with this character twice, two different varieties of that. So one second. So now we are now we are selfing the two plants. So now you have now you have a couple of gametes. Last time you only had two, like two gametes. So this time you're getting four. Like from each one of them you're having two each. But in the previous case, this had only one gamete, that is capital T, one type, okay? And this also had one type of gamete, small t. But over here you can see that each of one of them, each combination of alleles have two different gametes. Okay, so now we have the gametes over here. This is the gametes again. Okay, now we are forming the F2 generation. So these are going to fuse basically, and you get the F2 generation, okay? Now you can do this in two ways. I'll be doing both, okay? So we can do this in two ways. One is the Punnett square. Yeah, one second. So this is the Punnett square. Yeah, so this is one way to do it. Punnett square, and the other one is you can directly finish it from here. So I'll show both, okay? So everyone should remember genotypic ratio, so you can find it right now. So I'll be asking after this, okay? So first of all, this is the first type of offspring that can be formed. The second one is capital T and this one. Then you have this one and this one. And lastly, you have the small t and small t. Okay, Amanpreet, can you tell me what will be the phenotype of this one? You can see my pointer, right? Yeah, this one. Tallness, okay. Now, Marwan, can you tell me what these two will have? These two, capital T, small t, what will its phenotype be? Marwan. Okay, Shahid, can you tell then? Shahid, Ibrahim, anyone?
Which one?
This one. You can see my pointer, right? The laser thing.
Yeah. Tallness.
Yes, it will be tall because capital T is the dominant trait. And lastly, anyone can tell what will be the last one? Just anyone tell. Doesn't matter.
Shortness.
Short. So now you have three tall plants and one pair of short ones. So you can see that in the first generation only tall plants was there, but in the second generation just automatically you can see that a dwarf plant is there, even though you crossed to. Okay, now over here it seems obvious that like you can expect, okay, even though it's not formed, because you have a short plant and a tall plant. So naturally we would expect that a dwarf plant would be there, even though it's not formed, okay. Even though it's not formed, we can expect a dwarf plant, but over here you're crossing two tall plants and getting a short plant. So this happens because over here the gamete for dwarfness, the allele for dwarfness, it is included in the F1 generation, but it's not expressed because on the capital T, it's the dominant trait. It is basically nullifying the effect of the recessive trait. But when you're crossing this again, you find a homo. Okay, it's not written properly. 1 second. Yeah. So here you're finding a homozygous small t. So this is so both of the alleles are small t. So there is no dominance here. Both of them are the same thing. So it will be short. So you have tall, 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 and short. Dwarf. Okay, so this is one way of doing it. Now the other way, Punnett square. So for Punnett square, you have two different parents here, right? For Punnett square, you're considering this one and this one. So you basically take the two combinations that are to be selfed. Okay, so since this is the parent, so this is the father. Okay. Since this is the father and this is the mother, let's take the top part as father. So this is father and this is mother. Okay. So in father, this is father, right? So you can see two different types of gametes, capital T and small t. Oh, here also you have capital T and small t for mother. Now you just, yeah, what you have to do is, so in this box, you have to cross these two, the first one in mother and the first one in father. So you get capital T, capital T. And for this one, you cross the first one in mother, like you have to look the one which comes to your left. So here you can see if I go from left over here into the mother's side, you're getting the capital T, and over top in the father's, you're getting small t. Over here also, like from the top, you're getting capital T, and from the side, you're getting small t. And over here from the top, you're getting small t, and side also you're getting small t. So from mother's and father's side, you're getting small t. So basically, you're getting the same thing. From here also, you have capital T, the same four types you're getting, but it's just another way of doing it. So this is tall, these three are tall, and this is short. That's we have already discussed it. Now there's two more things: genotypic ratio and phenotypic ratio. So 1 second, let me just write over here. Oh no. 1 second. Genotypic ratio and phenotypic ratio. So can anyone tell what would be the genotypic ratio? Genotypic, if you remember. You can take a guess what would be the genotypic ratio over here, like in this case, the F2 generation. Anyone knows? Okay, so we didn't discuss this, so I'll just quickly tell it. Genotypic ratio, first of all, you have to write each and every one of them down, like all three. Like over here you have three different types, right? This two are one, basically this two are one, and this is another one. So you have three different types of genotypes. This is genotype, okay. This is a genotype, this is a genotype, this is a genotype. So you list all of them down. First of all, you have TT, capital T, small t, and small t, small t. You list all of them down. 1 second. Yeah. So now just put an equal to sign, and now look how many capital T's are there. You see that there's only one, so you write one. It's two. Now check how many capital T, small t's are there. You have one more. You have two. So you have two of them. So you just write two over here. Put one more is two, and look for how many small t are there. So you find that there's one, so you can just write one there. So this is the genotypic ratio. This is the genotypic ratio. Now phenotypic ratio. For phenotypic ratio, phenotype basically means, as I told before also, phenotype means the outlook of organisms. So after crossing, how do organisms look? Like from the character, we have two, right? Tall and dwarf. So over here you find that there are three tall plants. So if you're writing tall is to dwarf. So first you have to write the two contrasting traits equal to, then check how many tall are there. We have three tall, and how many dwarf are there? One dwarf. So this is how we cross. Is there any doubt? This is the phenotypic ratio. I think that's it. Yeah. So is there any doubt? No doubt, right? If there's anything you can tell, or I can move on. So can I go?
Yeah.
Okay, fine. So now I told about Mendel. So Mendel is the one that performed this experiment. So he made three laws. So two of them are related to this cross. So we'll just go through them. The first one is law of dominance. So law of dominance means that if you're telling in simple terms, then only the dominant trait can express itself. That's all that this law says. So here you can see that in the F1 generation, capital T, small t was formed, and capital T is the dominant trait. So only tallness is expressed. So that's what law of dominance says. The second one is law of segregation. So now the parents were capital T, and small t, small t. Now this capital T, it's not like glued together. It's not one. What do you say? So suppose if I'm, it's not one or attached structure. Over here, capital T, it's basically different genes, like different alleles. Okay, not genes. So it's not fixed together, but during reproduction, it splits into two and recombines. So here you see that capital T, small t has formed capital T, small t. So this is a different trait, right? Like this was not there in the parent, like this genotype. So what is the advantage of this is that it gives all different characteristics a chance. So this is not related, not much relatable to monohybrid cross. It's more relatable to dihybrid, because over there, like actually new types of thing like offsprings can be formed. Over here, it's basically only the parent and like the parent characteristics. Even though like in the first generation only tall is received, and in the second dwarf. So both these are parental characteristics, but in the case of dihybrid it all changes. So since there's no doubt, let's move on. So this is just the small way of representing it. So this is how we should do it. Okay, dihybrid cross. So dihybrid cross. Okay, now before this, one second. Before this, there is some reasons. One second. Let me just take the previous. Okay, sorry. So there is a couple of reasons why Gregor Mendel chose P plants. Okay, he chose P plants for all his experiments, both his experiments, dihybrid and the other one. So he chose P plants for both. So this was because the first reason is they have a lot of contrasting characters. So here we have one tallness, dwarfness. This is one contrasting character. There is also the color of flower. It can be white or purple. Also, it is another contrasting trait is the shape of the seeds round or wrinkled. So like this, there are a lot of contrasting traits. So this gave him a chance to do various experiments. The second reason is lifespan of pea plants is really short. Okay, so like 1 year you can grow two different sets of generation, like basically two different generations you can grow in 1 year. Now he can't test this on humans, because in humans he can only go through maximum 3 or 4 generations maximum, like, you know, right? Because he also has a time to live. He'll also die. So he can only test on 3 or 4 generations, like his all his experiments and stuff, to find similarities or a pattern of how the genetics works basically. So he could not perform on humans, but since he chose pea plants, a lot, like 2 different generations of pea plants can be grown in 1 year. So he got an opportunity to test different generations. Now another advantage is that, like, for conducting his experiments you need to be able to cross different plants, right? So over here we talked about the round and wrinkled seed, like all those experiments you need 2 different traits in the first place. So the plant has to have the ability to be cross pollinated. So pea plant is a plant that can be self pollinated as well as cross pollinated. So yeah, these are some reasons why he chose pea plants for his purpose, this experiment. So yeah. Okay, and now dihybrid cross. So dihybrid cross is a cross which has 2 different pairs of contrasting characters. So here we Mendel chose one second, not like this. One second, okay. So Mendel chose round green seeds and wrinkled yellow seeds. So we'll just do the cross. So the cross is like goes like this. So this is the parent, okay? Now the parent, okay, let's try this out, okay? Can someone tell me, now the parent characteristic is round green. Can someone tell me what, okay, I want to tell you, okay, first of all, someone volunteer. Can someone volunteer? I just have a small question. It's not that tough. Someone volunteer to come. Okay, I'll have to call. Badruddin, can you speak? Hello? Badruddin?
Yes.
Yeah, okay, so 2 different traits were taken. 2 different traits were taken by Mendel. Okay, the first one is round and green. So how would you denote round and green? By its genotype. So round is, I'll tell you this, round is dominant and green is recessive. So how would you denote it? You have 2 letters, okay? R,
Okay.
R, okay, that's an R, okay. Just like Y and Y, okay. These are the 4 letters you have to choose from. How would you make round and green? So you can just try.
So
Round is dominant. So
yeah, rounded dominant. Okay.
so I should take capital R or small r?
Capital R
Capital R,
and
capital R.
small y.
Small y, yes, small y. So this is our yeah, first parent. One second, let me just write it a bit more small. This one is kind of big. Capital R, small y. And the second one is the other two basically. So R and capital Y. So over here, capital R is dominant. So let's not consider that now, but basically it's round green. And yeah, small r denotes green, okay? So then you have small r, it is wrinkled. Oh god. Wrinkled, and the other one is yellow. So now before doing the cross itself, can anyone predict what the F1 generation would be? Just can anyone predict it? Just by looking at this. This is Y, okay? So can anyone predict it? What would the F1 generation be, the first generation?
Round and yellow.
Round and yellow, that's it. The first generation will be round and yellow. Why? It's because over here, first of all, we look at the capital letters. You know that in the first generation, the whole combination in dihybrid cross will have capital letters only. So here, when you cross, the gametes that you get will be, just erasing this, okay? The phenotype I'm just erasing for now because I can't do the cross then. So now over here, the gametes that will come, okay, what kind of color is this? The gametes is one is capital R, that is for round, and this is green, small r, okay, that's a small r. This is a small, no, that's small y, this is small r, and this is a capital Y. So these are the 4 different types, all are different gametes, none of them are similar, okay? So these are the gametes, so you can just write that down over here. So now let's see if what Badruddin told is right, okay? So this forms ry. So this is together, okay? I left a bit space over here. It's not supposed to be like that. ry, y, and ry. So they're just becoming one basically. From both of them, these two gametes are coming from the father. This is father and this is mother. So the capital R and small y are coming from father, and small r capital Y are coming from mother, and these are just going to combine. So you get R, and the other R, capital Y, small y. Just make sure that you're always writing the capital one first. So capital R you have, capital Y you have. So capital round Y, no, capital R is round, capital Y is yellow. So the phenotype of the first generation is round and yellow. Okay. So this is the one. Now you're selfing it. Okay. Now you're going to self it. Now when you're selfing, you get the same thing. Your selfing basically means, now suppose a plant has this genotype. Okay. Two plants have this genotype. You're just making them reproduce basically. You're just, that's what I told about cross pollination. You're setting the like environment for these two plants to only multiply or make its progeny. So that's what selfing is. These two plants cross in and when they cross, this is round and yellow, this is round and yellow. So when they cross, like in the previous method I told you a way where you can just put arrows and do it. This, if you do it like this, your page, there's no way you can complete it. So you have to do the Punnett square here. Okay, so just keep this in mind. I have to erase the screen. Okay. Okay, so I'll just write it down here. The last point, when you're selfing it, you're getting, when you're selfing, you're getting RR YY into RR YY. Okay, so for this, you need the Punnett square. Okay, you need the Punnett square. So four. Okay. Not very straight, but then yeah, make do. Okay. One second. Okay, so this is the Punnett square. Okay. Now, okay. So now, as I told, the same process will take it. So this is the father, and this is the mother. Father, mother means like their genotype. Okay, nothing else. It's just the genotype. So over here, there's nothing over here. Okay, it's not supposed to be there. So now for father, Kedarnath, can you tell me what to write here? This is the father's phenotype. So what should I write here? Just one by one. Can you tell me? Kind of separated like this. Can you tell Kedarnath? It's fine if you don't know. It's separated like this. Okay. Okay. Yeah. Kedarnath, can you tell anyone? Anyone can tell? What do I have to write here? Okay, then I'll tell. So over
If
here,
you write
yeah,
one
okay.
of them, write capital R,
Capital.
and the next one small r, then capital Y, and then small y.
Nice. So this is for father, and even mother is the same thing. So you just write the same thing down. Okay. Literally the same thing. Okay, now now you have to merge, like, what do you say, format.
Does the order matter?
Yeah.
Does the order matter? The order in which you put it in?
No, actually.
Okay.
One second. It's not like this. Not like this. Okay. There's a small thing. You have to do one more thing. It's not like one gene this time. You have to actually have pairs of it. Okay. One second. So this you do take it. Okay. I forgot about this. Okay. One second. Okay. You do take it like that, but then this is another way. Okay. So first of all, you list. Don't do it this in your paper. You can directly write it in the paper, but list all the types of gametes down, and make, now with 4 different types you can make, like, these 4 different allele like types, you can make 4 different types of pairs. Okay, pairs means just combine each of them, and each pair should have one R and one Y. So it's tough to say it, but you can understand when I'm doing it. Over here I took capital R, capital Y. Over here I took capital R, small y, small r, small r, small y, small y. No, small r.
I think you repeated one.
Yeah. Okay, so here both are capital. Okay, this is both are capital. Over here, capital R, small y, small r, capital Y, small r, small y. Next one, the same thing, capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. So these are the 4 different pairs you have. Okay, now we can do it, because you can just, again, you have to look, if you're in this box, you have to look what's like on top of you and what is beside you. So here, if you're doing, you have, first, again, you have to write the same letter together. You have two R's, you have to write both of them together. You can't write it in different places. So in that sense, order matters, but otherwise order doesn't matter. Okay, you can write this in any way you want to, but then in the sense you're writing, like when you're writing this, order matters. So yeah, so here you have both capital, but one Y is small. Okay, this is gonna take time. Might take time.
Is this pairs of genes?
Yeah.
Is this like pairs of genotypes?
Yeah. See. In these pairs, dihybrid pairs, both the characters will be there in one, like, combination. So that's why I took both over here. Like in each, like, combination, you'll have both of them. So that's how the cross takes place. So the cross is mainly between 2 different, like, when you're crossing, you have both 2 different, what do you say? Both of the different contrasting traits together, the different ones. Okay, not the same contrasting trait, but one from each contrasting trait. I'll explain it. Okay, let me just write.
Okay, fine.
So it's better you know the ratio, because just in case if we get this wrong. Last one. Yeah, done. Yeah. So we took combinations over here, like different, one contrasting trait from the, like, 2 different contrasting traits we had, and we crossed them. So there is 1, 2, 3, 4, 8, 12, 16. So you have 16 different possibilities. Okay. So now we'll do the phenotypic and the other ratio, but can anyone tell which one is wrinkled and green? There's only one case here that is wrinkled and green. Which one of these is wrinkled and green?
The first.
First one. Okay, I'll just. Oh no! One second. One second. Yeah. Oh no. Where is the empty one? Yeah. So I'll just remind that capital R is round. Capital. Oh my God. Capital R is round. Capital Y is yellow. Small r is wrinkled. Small y is green. So now tell which is wrinkled and green.
It will be capital Y and small r, right?
Wrinkled and green, okay? Y and no. Yellow and round are dominant because yellow has capital and round is capital R. Wrinkled is small r and green is small y. So what would be wrinkled and green offspring? Which one of these?
So small r and small y.
Yeah, this one, right?
Yep. Yeah,
Yeah,
last one.
this is the only wrinkled and green. Now, another thing is, if you remember from the beginning, the parents, what was the parents? What were the parents? Anyone remembers? Like, what were the genotypes of the parents?
Round and green.
Wait. 1 second.
Round and yellow.
And
Round and yellow.
then we have this one.
Hello, Aman.
Yeah. Yeah.
In the starting, you said that. Wait 1 second.
Yeah. You can ask anything. It's fine. Anything. That's the point of this. These were the genotypes, by the way.
Yeah, in the starting, you said capital R and small r was round and green.
No. Capital R and small y. Okay.
No.
Okay. Let me just explain that then. Okay. So this, you can see my screen. I'm writing down. This is one pair of contrasting characters. This is one allele. This is another allele. So the capital R allele represents round. Small r allele represents wrinkled. So this is allele based on shape. Like, yeah, the characteristic based on shape, not allele based on shape. So over here you have capital Y, that is yellow, and small y, which is green. It's another pair of contrasting characters. And over here we consider two parents. Okay, now the first parent had both capital R, this is round, and two small y, which is green. So we have round and green is the parent, first parent, father. Now for mother we had small r is wrinkled, and capital Y is yellow. Okay, so this is the phenotype of the, like, the parents. Now in the first generation we got capital R, and small y, round and yellow. Okay, so from the father, like from the parents only had round green and wrinkled yellow. But when you're crossing them, you're getting two new stuff. Okay, like two new different types of plants, or yeah, characters. The first one is round and yellow. You can see round and yellow over here also. Round and yellow and wrinkled and green. So you're getting two different characters that never existed in the parents. Like it never even existed, but then it came because like it was there in the parents. Like the chances of this was there in the parents, but then it wasn't in the correct state. You have to get it in that correct order to have the characteristics appear. So for example, if you see, as we told, round and green was the parental characteristic. So first of all, let's mark the parental stuff over here. Okay, I'm marking all the parental ones with green. So the parental first one was round and green. Okay, so this is one round and green, this is one round and green, this is one round and green, and the other one was wrinkled and yellow. Wrinkled yellow, 1, 2, 3. So out of the 16, only 6 of them have parental characters. The rest of the 10 are new. They're not differently new, because most of them are same. Like there's basically only two. There are two different new types of plants that appeared apart from the two parental ones. So that is the advantage of this. So this is, I'm just going to tell all of them. This is round yellow, this is round yellow, this is round yellow, this is also round yellow. It's because each one of them has a capital R and capital Y. And if you have both capital R and capital Y, you don't need to look at the others. It will 100% be round and yellow, because these two are dominant. The next one, this is also round and yellow, but this is round and green since you have small y. Both are small y, homozygous. This is round in yellow. This is also round in yellow. You have capital R, capital Y. Over here you have capital R but small y. So this will also become round in green. We marked it here. This one is round in yellow. Another one round in yellow. This is also round in yellow. This is wrinkled in yellow. Both are small r's and both are capital Y. So this will become yellow, and this, because of this, we have wrinkled. This is wrinkled yellow. You have two small r's, then 100% it's wrinkled. And you have one capital Y, 100% it is yellow. This is also round in yellow. This is round green, wrinkled yellow. And the last one, since all of them are recessive, it's wrinkled and green. So this is wrinkled and green. Is there any doubt here?
No.
Okay, so last thing there's two things actually. Okay, so now let's write the ratios. So here we only have the phenotypic ratio, because the other one, you can't do it, like it's not that easy. So we'll only be doing the phenotypic ratio. No, we're only learning phenotypic ratio first of all. So yeah, phenotypic ratio. So let's count the, so basically, you have, okay, I'm not writing it down right now, but then round yellow, I'm just writing it down like this, okay? Round yellow is equal to round green is equal to wrinkled yellow is equal to wrinkled green is equal to, I'm just writing the in genotype form. But when you're writing, suppose if you're writing for some exam or something, you have to write round yellow. Just write it in words. Round yellow is equal to number. So number we'll find out now. So this is round in yellow. This one, we're starting from here, okay? This box we are considering right now. Okay, round in yellow. Okay, so we have one round in yellow, we have two round in yellow, 3, 4, 5, 6, 7, 8, 9. Okay, so we have 9. Next one we are looking for round and green. Round and green. 1, 2, 3. Next one is wrinkled yellow. We have 1, 2, 3. And the last one, we only have one case of wrinkled and green. So the ratio you're getting is 9:3:3:1. And if you count them, you're getting 9 plus 3 plus 3 plus 1 is 16. So there are 16 different types. If you count them, you can get it. Is it fine? Yeah?
Yeah. Yeah.
So another thing now about genotypic ratio. Now, if you're looking here, all 16 of them are different, right? Each and every one of this is different. So calculating genotypic ratio, it wouldn't be smart, because first of all, it's really tough, because over here when you're calculating TT, like the monohybrid cross, you in the end you're getting TT, capital T, both capital and both small. You only had 3 different types. Over here you have 16 different types, so you can't do that. So you're only considering the phenotypic ratio. So that's it for that was dihybrid cross. Okay, so there is a law, another law, last law. Okay, so the law here is that, see, when 2 or more characters are inherited, 2 or more, so by hearing the 2 or more part itself, you understand that this is for dihybrid. Okay, so when 2 or more characters are inherited, individual hereditary factors assert independently during gamete formation, giving different traits and equal opportunity for occurring together. So this is the statement. So now let's try to understand it. So here, parent, I lost the parent. So the parent was capital R, both capital. The first parent was capital R, small y. Second parent was small r, capital Y. So I just told that these are 2 different traits, right? So when you cross them, like it's not, you can't expect the same child, like same offspring, because they're not one. Like, let me just write them down. One second, let me just clear the slide. Okay, this was what we considered right now, right? So this is what we considered. So this set, right, this full set, it's not stuck together. It's kind of like how we discussed in the first rule. It's not stuck together, or it's not structurally like merged. Like in its nature, it's not merged. It separates out, like in many ways we told, like capital R, capital small y, small r, small y. This one, like this, it can separate. So first, his point was that they are not sticking together. So what is the advantage of this? So you remember we wrote this down, right? Like in a Punnett square, we had written like this, right? So we got 16 different types of genotypes from just two different types of genotypes, right? So basically the fact that they can separate during gamete formation, it gives different characteristics a same and equal opportunity to occur together. So that's just the statement that he had told. So it's just that in the end, when you're having like when the gametes can separate from each other, so when these combine together during gamete formation, it gives different traits a different chance. So in this case, you saw that round and round and round and wrinkled and green no round and wrinkled no round and green and wrinkled yellow was the parental characters. But in the end, you got like in the end after you cross them, right, in the second generation you got wrinkled green and round yellow. So these were not even included in the parental or like what do you say set. So that's just what the fact like that's what he said. So you can see here that like same thing, okay, literally the same thing. This is the phenotypic ratio, this is the Punnett square, and yeah, this is just a pictorial representation. So you can see that the parent was round and green, the other parent was wrinkled and yellow, and in the end you got this one and this one. These two are new ones. So yeah. Okay, so you can see my screen, right? Am I visible still now? I think I stopped screen sharing, right?
No, we can still see your screen.
Okay, one second. Okay, so law of independent assortment. It's independent assortment. Same thing, when two or more characters are inherited, individual hereditary factors assort during gamete formation, giving equal different traits and equal opportunity of occurring together. So this is the same thing. Okay, now okay, before this. Okay, so before this, there is another thing. 1 second, okay. Okay, so blood groups. Let's just quickly do it. Okay, it's not that important, but then let's do it anyway. Blood groups. Okay, so we know that there are 4 different types of blood groups. We're not considering negative and positive now, okay? It's not there. So blood groups are of 4 types. So you have blood A or blood B, blood O and blood AB. So when you have A and A together, like suppose you get A from the father and A from the mother, blood A. So in this combination, it will be blood group. The offspring is going to get blood group A, because it doesn't matter if A is recessive or dominant. It will get, because both of them are A, right? Now another thing is that before I go on, A and B are codominant. Okay, codominant means we talked about dominant and recessive genes till now, but both of them are dominant. So when you have I is how it's denoted. Okay, blood group. So blood group is denoted by I. So when you have this, both of them will, like, you're considering both, because none of them is recessive, none of them is dominant. They're both codominant. So this is why you have a blood group AB. So this is blood group AB. This is blood group A. Next one is O. Okay, so right now I'm telling you A is dominant. I told you A and B are dominant, codominant. So what would this be? Which blood group with this form? So father donated, or father basically gave blood group A to the offspring, and mother gave blood group O. So what would the blood group of the child be? Anyone? A or O? Just two options, A or O. Anyone can tell? Just have a guess. A is dominant. Okay, so Amanpreet, can you try? I'll just call out then. Amanpreet.
Uh,
Yeah. So blood group, this is the genotype of the sun in blood group, okay? So A is dominant, O is recessive. So what will the blood group of the sun be? The progeny. Blood group A or O?
yeah, the blood group of, I think it'll be
A, right? It will be A because A is dominant. See, whenever you have either A or B in your blood group, 100% it will be A and B. Except in this case where it will be both A and B because A and B are co-dominant. Both of them are dominant, so it's equally dominant. So both of them can show their characters. Last one is so this now if you want to, if you have O blood group, then both of your blood, like both your father and mother has to contribute O blood group. So this will be O, and the last case is B. So B also. It can either be like this, it can also be like this. Both cases you'll get O because I mean B, because B is positive. Like not B is dominant. Okay. So now there's another thing like okay, yeah, but I think that's enough for blood group right now. For that, for now, it's enough. It's not included anyway. Okay, so next one. Okay, one more thing. So see, like, it's kind of related to the last chapter, but it'll help you understand a bit more better. Now, when you have characters from parent and like father and mother, okay, just imagine this is one, okay. So if there is, okay, let's just leave this, okay. 46 chromosomes. Okay, this is chromosomes, okay? This is chromosomes. So 46 chromosomes from father, 46 chromosomes from mother. So no, everyone has 46 chromosomes, 23 pairs of chromosomes, okay? But when when during gamete formation, your gametes will only have 23 chromosomes because they go through meiosis. Like during gamete formation, the number of chromosomes in cells decrease, okay? So like decrease means it reduces to half. Now, why does this happen? Now, suppose this never took place. This never happened. This imagine this never happened. Okay, so so father has 46 chromosomes, mother has 46 chromosomes. When the gametes are fusing, you have 46 plus 46 chromosomes. So how much will that be? 92 chromosomes in son, right? Correct, right? Yeah, so the son will have 92 chromosomes. Now, again, if this happens, 92 chromosomes in father, 92 chromosomes in mother, when these come together and form another child, you'll get 184 chromosomes. Okay, so do you think this is right? It is right. Like father, in the beginning, father has 46 pairs of chromosomes, and when their offsprings are formed, is becoming 184. So this is wrong because it will, first of all, it's not possible. Second of all, if it was possible, it would cause mutate, like a lot of problems basically, because you're only supposed to have 46. That's just the number. It's like there's a limit to stuff, right? So over here, what happens is that 46 chromosomes for father and 46 for mother. During gamete formation due to meiosis, we learned last year, it becomes half and it forms 23, 23. So when this is the, like suppose if it's father, then it's the sperm of father. This is the egg of mother. And when these combine, you get 46 again. So you can see that the chromosome number is restored. And two different sets of characters are coming from father and from mother. So variations will also be produced. So we are going to look into that. Okay. So, okay, now before that, so different species use different methods of for reproducing, no, for determination of their gender, okay? So first we'll be talking about environmental, like how environment affects the determination of the gender of an individual in of an organism animal. So in some animals, the incubation temperature determines what the gender of their offspring will be. Incubation temperature is basically the temperature at which the egg hatches, okay? Egg means like birds and stuff, eggs. So yeah. Okay, okay, I'll give some examples of this. One second. In turtles, the fertilized egg, again, even in turtles, like the temperature at which the egg is kept determines the gender. Now if the egg is kept at a high incubation temperature, then female turtles are produced, and if it is kept at lower, then male offsprings are produced. Like another opposite example is of what do you say, lizards. In lizards, high incubation temperature produces male offsprings, it's opposite, and low will produce girl offsprings. And there's another one, snails. Snails is completely different. They can change their gender whenever they want to, because it's determined by environmental factors, but then they can change their sex, which determines that it is not genetically determined. But in humans, the like the gender of the individual, the sex of the individual, it is determined largely like by genetic means. We know it's all about genetics in humans. So the genes that is inherited from our parents will decide whether the offsprings will be boy or girl. So that is the different strategies. Now, how do traits? We'll get back to this. We'll get back to this. Okay, so first let's do the cross. Okay, we'll do the same cross. So now, okay. Let's go back. So as I told, human beings have 23 pairs of chromosomes. That is 46 chromosomes basically. In this 26 pairs, 22 pairs are called autosomes. Okay, autosomes means the other chromosomes, like the chromosomes that is not involved in any like sexual factors, like it won't determine any of your whatever changes that happen during puberty and stuff, all that, it's not determined by autosomes, but it is determined by the last pair. It is called sex chromosomes. So this 23rd pair is responsible for all your traits that is related to this. So in males have two different types of chromosomes. One is X and one is Y, but female only has one type. It's both X. Okay, both X. So now let's do the cross. So as you can see over here, it's written out of these two chromosomes, female has both X and male has one X and one Y. So sperms has now. Okay, now in sperms also there's 50% X and 50% Y. So it is an equal chance. It's not unequal. It is an equal chance since both of them are half. Okay. Now it's written over here, some sperms have X and some has Y chromosomes, but all of the eggs has X chromosomes. That is, all female gametes carry only X chromosomes. Now if a sperm carrying X chromosome fuses with a girl egg having X chromosome, then the chromosome, basically the what do you say, the genotype will be XX, and XX means that the child will be a girl. And if a sperm having Y chromosome fuses with an egg having X chromosome, then the child will be a boy because the genotype formed is XY. So let's do the cross. That's basically it. It's the same thing we learned in 8th. Anyway, let's just do the cross because we didn't do that yet. Okay, so the cross is from male, female. Yeah. Okay, over here you can see that when X and X fuses, you get XX. So these are two. This is the first one and this is the second one. Both of them are girl. Now over here, you have XY. When our X chromosome fuses with Y, so now you'll get a boy. So can someone tell me what is the percentage of getting boy and girl? Someone tell me. Badruddin.
50.
50%. So you have a 50% chance of getting a girl and 50% chance of getting a boy. So this is because, first of all, the male determines the Y chromosome. In specific, the Y chromosome in male determines what the gender of the child will be. And another thing is that, like, what do you say? How do you say? Yeah. So yeah, that's it basically. The male determines which gender the child will be, and also the sperms that carry X chromosome and that which carries Y chromosomes, it is equal. So 50% carries Y and 50% carries X. So even this is half. So in the end, 50% for male and girl, for boy and girl. So that is what this is about. And the last subtopic now. This thing is our last topic now. It is how do traits get expressed? Okay. Okay. So this one is bit different from the whole chapter. Now, DNA is the information source for making proteins. We learned it in the last chapter. Now, gene is a section of DNA that provides information for one protein. Okay. Now, this is basic. You can understand this with an example. Okay, mainly. So, okay. So there is a plant. Okay. So this plant, it is reproducing. Okay. So we have two plants. It is reproducing, and it forms a plant which has the genotype TT, okay, TT, okay. So what is the why? Like, can anyone tell what its phenotype is? Like, what? How does it look? Dwarf or tall? Kedarnath, anyone can tell? This plant with genotype capital T, does it look short or tall?
Tall.
It looks tall, yeah. Yeah, it is tall because both of them are capital. Now, how does it become tall? Like in the beginning, the genes are tall, okay? The gene it gets from the parents are tall. Let's see how this happens. So, you know about the growth hormone. So these genes, like it has hormones that can trigger growth, okay? So this is a plant, right? This is a plant, not human. So this it releases hormones that can trigger growth, okay? So plant height depends on how much hormone is released, okay? Now since this has capital T, we know that it's tall. So whether a plant is tall or short, it depends on the amount of plant hormone, like the efficiency of the process for making this plant hormone, okay? Now an enzyme, so growth hormone in the plant, the growth hormone, if this or like enzyme it works efficiently, then a lot of hormone will be made, growth hormone. And if a lot of growth hormone are made, then the plant is tall, we know that. On the other hand, if the genotype is like this, then the plant will be short. So the enzyme action, it is less efficient, and since it is less efficient, the plant like less growth hormones will be made, and hence the plant will be short. Okay, that's it. So genes control the characteristics or traits of human beings or organisms, any sort of organism. And this is just tallness or shortness, but the same process is there for all other types of characteristics as well. It's not just tallness or shortness. Okay. Acquired traits and inherited traits, you can understand the meaning of this from the name. Acquired traits are acquired during one's lifetime. Inherited traits are what you get from your fathers or your predecessors, basically. So you can't inherit, like you can't give your acquired traits to your offspring, but you can give your inherited traits to your offspring. Now acquired traits are not included in the genetic make. Okay, we'll come back to this point. Okay, now suppose, let's take an example. Okay, so a person is good at singing. Okay, what is it a inherited or an acquired trait? Can anyone tell, Badruddin?
Acquired trait.
Acquired trait. What about tallness?
Inherited trait.
Okay, so now singing. So just because you develop singing, you went to singing classes, okay? You have, like, in your childhood, you have gone to singing classes and you have acquired this trait to sing properly, and you're really famous for it, okay? So just because you can sing, that doesn't mean that your offspring can sing, okay? So it's not inheritable. Also, it's not okay, genotype, okay? Now genotype. So you do not have a specific genotype called singing. Like, that's not a genotype. So since this is not a genotype, it's not going to get transferred to your offspring. Also, the change in DNA, it does first. Okay, leave this line. Okay, but basically it means that it's not going to affect your DNA. Like your character, the trait of singing, it has no impact on your chromosomes, that is sex chromosomes. Basically, your DNA, it has no impact on it. So change in DNA will not result in such genes. Okay, so we know that like traits are created by variations, but variation does not create singing. Like that's just an acquired trait. On the other hand, inherited traits. Tall. Let's consider tall plant. It is inherited from predecessors. For example, when you are selfing capital T, small t, you're getting when these come together. Oh, they move this. You're getting capital T. So it is inherited from predecessors. These are inheritable. Over here, you can see this is in these two are coming from the parents, and it's inheritable basically. It is present in the genetic makeup. So this is a genotype. This is the genetic makeup. So that's I'm just giving an example over this. And a change in DNA will bring about in such a change in traits. We did the cross, right? When you're crossing this, you get this. And when you're doing this again, when you're selfing this, you're getting this. So first it was capital T, then it became capital T, small t, and in the end it came back to this. So there was a change in DNA at this stage, and that is the reason why it brought about such a change. And again, another change took place which brought back the parental character. So it's just that, it's not that important. So yeah, this is the last thing. Now we have 2 questions. After that, we'll do the test. Okay, I'll just quickly finish off the 2 questions. Okay, anyone volunteers just to answer? I'll help you. Anyone? Badruddin, can you come? Just try it out. You can try, Badruddin. Okay. I'll write this stuff for you, okay? Now you have a purebred recessive dwarf. You have dwarf and you have tall, okay? So when you see that, you just directly write this stuff down. You don't need to do it if they don't ask you, but still you can. I'm just writing just to explain it, okay? TT, so this is the F1 generation. And now you're selfing it. And you're getting T, 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 and TT. Okay, yeah, Badruddin, you can tell now. What do the plants in F1 generation look like? This is one. F1 generation look like. This is the F1 generation. What do they look like? Yeah? Hello?
It will be tall.
It will be tall. That's the answer. Good. Okay, second one, state the ratio of tall plants to dwarf plants in F2 generation.
Should I answer that as well?
Yeah, you have to answer the full thing.
Okay, so it will be 3:1.
3:1. Basically, it's the phenotypic ratio. Third one, state the type of plants that is not found in the F1 generation but appeared in the F2 generation.
The dwarf one.
Dwarf one. So this is, you just need to write like this. So this is how you write the answer. You just need to write it small. So that's the good part of this chapter. You just need to write it like this. Next question, Kedarnath, can you try? With the help. Okay, fine. So you can control the screen, so you can do it. Just I'll enable annotation. Okay. So now you can draw the thing. Just draw it. I'll tell. We already explained it basically. Okay. So first of all, you tell then who is responsible for the sex or gender of the offspring, father or mother?
It depends.
Yeah. How?
It depends on the
the
gene.
father. Right?
It's the father. See. Father is XX, no, XY, and mother is XX. So what determines this gender? This Y chromosome, only this Y chromosome of the father determines the gender. Mother has both X, right? Mother has both X. So first of all, 2 possibilities is X, Y, and XX, the child, okay, when these cross. So over here, if father contributes X and mother 100% X only. So if father contributes X and mother contributes X, you get XX, which is a girl. And if father contributes Y and mother contributes X, again mother is X, you will get a boy. So only these two, there's only 2 possibilities. This is 1, this is 2. So you can conclude that father is responsible for the determination of the gender of the child. Now can you draw the flowchart? That's the same thing. Flowchart means this only. Yeah, can you? Kedarnath.
I don't think I can. I'm trying to draw, but then I can't draw it.
Oh, why? I enabled it, right? You can try now. Anyone can try? Let's try
I
drawing.
can zoom in, but then I can't draw.
Try. Anyone else can try? Anyone can do it, then just try. Okay, someone is drawing. Who's this? Ritvik, are you joining from 2 accounts? Okay, fine. So look, it's the same thing. You just have to do it like this. This is father, this is mother, this is mother. Father gives this, mother gives this, father gives this, mother gives this. So when you're combining, you will get XX, XX, XY, and XY. I find it XY. So this is boy, boy, girl, girl. Okay. No, it's girl, girl, boy, boy. XY is boy. Okay. So that's that. So this is the chapter over, but then I have a small test. Okay. 1 second. So ma'am wants the marks because this is the only evidence we have. If you have any doubt, you can ask. So you can go on. I'll post it in the Zoom chat. Okay, you can take it from there. Yeah, you can take it. I posted in the Zoom chat. You can all do it. Can you see it? Can everyone see the thing? Hello, can everyone see the link?
Yeah.
Okay, fine. So that's the chapter over. Now you can do the test. So yeah, that's it.